Okay, we're going into chapter 4 today, cell structure and function. We've talked about cells previously in other chapters. Today we're going to get into detail. We said earlier in chapter 1 that the cell was the smallest unit of life. I hope nobody missed that on the exam. Cell is the smallest unit of life. That's where we see those 5 characteristics of life emerge. Most of you know that most cells are microscopic. Most cells are microscopic. There are actually some cells that are not microscopic. Anybody give me an example of a cell that's not microscopic? A frog egg. Ever been out in a pond, seen that gooey mass of gelatinous frog eggs in the pond? Those are actually cells. Okay? So cells, the smallest unit of life, and most of them are microscopic. You may think that this amoeba here looks like just a blob, but this is a one-celled protist. Maybe we'll see some in some pond water later on when we get the microscopes out. But if we were to look at this amoeba and study it, we would see that it's very organized, and it actually exhibits all of those 5 characteristics of life that we talked about in chapter 1. It's organized. It acquires material energy. It responds. It reproduces, and it adapts. Okay? Some cytologists, a very famous cytologist that we need to mention before we go on, a gentleman by the name of Schleiden was a German who said back in the 1800s that all plants are made of cells. All plants are made of cells. Now this seems kind of silly to us today, but back then it was not silly at all. They didn't know that all things were made of cells. So this was a revolutionary statement that all plants are made of cells. And if you notice, I've got an illustration there of an Elodea plant. And I put that illustration up there on purpose because we're actually going to be looking at the cells of that plant in our lab number 3. You see that it's a long strand with lots of little leaves there. We use this plant in lab because those leaves right there are only 2 to 3 cell layers thick. So it's really easy to see the cells. If you'll notice back in the aquariums in the back, I have lots and lots of that plant back there. It's called Elodea. You can almost think of it kind of as an aquatic kudzu. Okay? Very prolific. Another gentleman by the name of Schwann said that all animals are made of cells. About the same time. Again, revolutionary statement. Schleiden and Schwann. Sounds like a poker group, doesn't it? Schleiden and Schwann. The way that I've always remembered that, Schleiden and Schwann, is that Schwann sounds a whole lot like Swan. So he did animals and Schleiden did plants. Okay, so Schleiden and Schwann basically said all living things are made of cells together in their word. Another gentleman by the name of Virchow, very, very famous cytologist. Cyto means cell. Okay, very famous cytologist, said every cell comes from a pre-existing cell. Every cell comes from a pre-existing cell. Virchow. Every cell comes from a pre-existing cell. I think that's kind of awesome to think about. Every cell in your body, millions and millions and millions, billions of them, every cell in your body had to come from another cell. Wow. Every cell on the planet came from another cell. That should lead you to that age-old question, which is, somebody, where did the first cell come from? Exactly. Okay? Should give you something to think about. So, when we put all 3 of these gentlemen's work together, we come up with something that's known as the cell theory. Now remember, in science, a theory you the connotation that we don't know if it's true or not, right? But in science, it's totally opposite from that. If I say something is a scientific theory, remember back in chapter 1, we said that is a concept that's supported by lots of experiments and observation of lots of people's work. So it's a given. We commonly accept it as being a truth if it's a scientific theory. So the cell theory puts all three of those gentlemen's work together and says basically that the cell is the fundamental unit of life. All living things are made from cells. And all cells come from pre-existing cells. And that is the cell theory. The cell is the fundamental unit of life. Cells, all living things are made of cells, and cells come from pre-existing cells. So just how big is a cell? Let's take a look at this little chart to see how big cells are relative to other things. And go back to your information that we looked at in Lab 1 when we talked about metrics, and we learned what a meter was, and a centimeter, and a millimeter, and a micrometer. Remember that? So let's take a look at these images. And we see over here on the right side, we're measuring things in meters. The tree is 100 meters. And we go down and it gets smaller and smaller and smaller, obviously. Down from meters to centimeters, millimeters, and remember the U with the funny little tail is a micrometer, okay? So you see above here, you see the measurements here that we can see with the light microscope and the electron microscopes. Electron microscopes are much more powerful than light microscopes. We have light microscopes here at Wallace. You have a compound light microscope. So let's take a look at this for just a moment. We see we're going with the frog egg here at 1 millimeter. We go down from that, and now we're in micrometers. And if you remember, a micrometer is 1,000th of a millimeter. Remember that? 1,000th of a millimeter. We can see most animal cells and plant cells in the light microscope. So you see right here that plant and animal cells are between 10 and 100 micrometers. Right below that, you see bacteria, and then you see mitochondria and chloroplasts. Those are compartments within cells that we're going to call organelles. We're going to learn what those are today. And you see that those are about 1 to 10 micrometers right there. So this is what we can see with our light microscope. This is where cells would fall, right there. Looking on down to the things that are smaller than that, you see the virus is extremely small. It's actually measured in nanometers. The nm is a nanometer, and that's 1,000th of a micrometer. Very small. Below viruses, we see those organic molecules that we studied in chapter 3, like proteins and lipids and carbohydrates, etc. And then on down in the single-digit nanometers, you see the very small molecules, and that is an illustration of water right there that you can actually see in the single nanometer. So just to give you an idea of the relative sizes of things and where cells fall within that. Okay. Ever wondered why cells have to be small? Has anyone in here ever seen the movie The Blob? Ever seen The Blob? What was The Blob about? It was a blob. It started out very small. It was a blob, and then it started eating things, right? And then it got bigger and bigger, and it was still a blob. And then it started eating people, and I think eventually it ate New York or something, didn't it? I mean, it was just this giant blob. Well, I'm here to tell you today, and I'm going to explain why that could never happen. Okay? The blob is definitely science fiction. You don't have to lose sleep or worry about the fact that some giant cell blob is going to come eat Huntsville, Alabama. Okay? So, why does thing very important to a cell is surface area to volume ratio. So let's think before we get into this. Exactly what are we talking about when we say surface area to volume? What is surface area? What's the surface area of something? Anything? What's the surface area? The covering. If I'm going to give you a Christmas present, let's say I'm giving you a box for Christmas, and I wrap it in Christmas paper, then it would be how much paper it would take to cover the box. So the surface area is the covering of something. It's the amount of outside. Okay? So for a cell, what is the surface area of the cell? It's the membrane that covers the cell. It's the plasma membrane. Okay? So the surface area of a cell is the plasma membrane. Very important to understand that the plasma membrane has a job to do. Very important job. As a matter of fact, we've got a whole chapter, chapter 5, dedicated to what the plasma membrane for the cell actually does. Okay, so it's the surface area. It's the job of that membrane. It not only separates the cell from the rest of the world, but it's the job of that membrane to control what goes in and out of the cell. We actually call it selectively permeable. So it literally will let some things in, but there are other things that it won't let in. So it controls what goes in and out of the cell. Everything that goes in the cell has to go through the membrane. Everything that goes out of the cell has to go through the membrane. Okay? So, volume. We're talking about surface area to volume. So let's talk a little bit about what volume is. What is the volume of something? Talk to me. Huh? How much is in it? That's exactly right. So the box that I'm giving you for Christmas, if I fill it full of sand, okay, I'm giving you a box of sand for Christmas, then the volume of the box is how much sand it holds, right? Well, for the cell, what is the volume of the cell? It's everything that's inside the cell. It's the contents. And remember, the contents are alive. It's not just a box of sand. Those contents are alive. They have to have nutrients. The contents of a cell has to have oxygen. It metabolizes and produces waste products. Those have to be released from the cell. Everything that comes in has to go through the plasma membrane. Everything that goes out has to go out. But the contents of that cell has to have oxygen, it has to have nutrients, and it has to release waste products. So the volume of a cell is the contents of that cell. And it has to be maintained, or it dies. So, let's take a look at this cube. And I've used a cube because the geometry is easier, okay? And there actually are cells that are cuboidal, that are cube-shaped. There are some that are rectangles. I use a cube as an example just because the math is easier, okay? We're going to take a look at this surface area to volume concept and see what happens to a cell as it gets bigger, okay? So let's calculate the surface area of these three cubes. And obviously, you can tell by looking at them. They're getting bigger, right? The first one is 1 millimeter cube. The second one is 2 millimeter cube. And the third one is 4 millimeter cube. And you can tell that it's getting bigger. But let's take a look at the surface area. In geometry, those of you that are math with this, in geometry, how do we calculate the surface area of a cube? Anybody? Geometry? Mr. Smith and Mr. Boyd are watching you. Two sides, length times width times how many sides? 6. So it'd be length times width times 6, because we have 6 sides. So basically we're finding the area of a square and multiplying it times 6. Okay? So 1 by 1 times 6 is 6 millimeters squared. 2 by 2 times 6 would be 4 times 6, so that's 24 right here, millimeters squared. And And then the third cube is 4 by 4 times 6, right? Everybody in agreement that that's the surface area of those cubes. That's how much wrapping paper it would take to cover. Okay, so obviously it's increasing. Well, we saw that. We know that. It's going from 6 to 24 to 96. Okay, let's take a look at the volume of all three cubes. Volume is a little bit easier. That's the answer most people give when I ask the surface area. But to find the volume of a cube, it's length times width times height. That's exactly right. So let's do the volume of this cube. The first one is 1 by 1 by 1. So that's 1 millimeter cubed. The second one is 2 times 2 times 2. So that's 8. The third one is 4 times 4 times 4. So what's that? That's 64. Well, obviously it's increasing. We know that. But what's important to the cell? Keep in mind the surface area has a job to do. What's important to the cell is the rate at which they're increasing. Are they both increasing at the same pace? And the way that we would look at that is look at them as a ratio. And we're going to put the surface area on the top of the ratio, the fraction, and the volume on the bottom. So we're going to first look at 6 over 1. Surface area to volume of the first cube would be 6, right? Surface area to volume of the second cube would be 24 over 8. Well, when we reduce that, that's 3. Surface area to volume of the third cube is 96 over 64. Reduce, that's 1 1/2. Notice that those numbers, 6, 3, 1 1/2, as the cube got bigger, the surface area to volume got what? Smaller. That's exactly right. Think about it as a fraction. For this number to get smaller, what's happening to the fraction? You've got a numerator on the top and a denominator on the bottom. And if that number is getting smaller, that means the denominator is getting bigger faster. Right? Because if the numerator, the number on top, was getting bigger faster, the number would be getting bigger. Surface area to volume ratio would be getting bigger, but the number on the bottom is getting bigger faster, which is causing that ratio to get smaller and smaller and smaller. So what does that mean for the cell? That means the volume is growing at a faster pace than the surface area. Well, the surface area is what's supplying the oxygen, the nutrients, etc. to the cell. So at some point, the covering of the cell is not going to be adequate enough to supply enough oxygen for the entire contents. So if we start with a cell right here that's small, and it grows and it grows and it grows and it grows and it grows, and it gets so big that now the surface area can barely support the volume of the cell. That big cell has two choices. It can either die or divide into two smaller cells so that you get that surface area to volume ratio back to normal. So then you end up with two little cells that grow and grow and grow and grow, and they both get large, and they have two choices. They either die, they have to die, or they have to divide. It can't grow anymore because the contents of the cell would start to die. Okay, that whole scenario, the time between when the cell divided and was small until it divides again, we're going to call that the cell cycle. We got a whole chapter devoted to that. Okay? So, has everybody got it? Surface area to volume ratio is extremely important. For a cell, it has to be large. That number has to be large. Okay? And we know that the surface area to volume ratio gets smaller as the cell gets bigger. And at some point in time, there's not going to be enough surface area to supply oxygen, etc. for the cell. So cells need a large surface area to volume ratio. Got it? Okay. Another reason that cells have to stay small. The control center of the cell is the nucleus. The control center is the nucleus. The nucleus literally controls everything about the cell. This is where the genetic information is. And there is only so much cytoplasm that one nucleus can control. Throwing some slides up for you to see the nuclei of some cells. Got kind of an assortment, got animal cells, plant cells. The first one you're looking at there is a plant cell. That's onion cells, and they've been stained with a stain called methylene blue. There is the nucleus of those cells. Oftentimes when we are looking at cells, we use stains. When you're looking at a cell that is unstained, you can't see much of what's inside the cell. Usually the nucleus is kind of translucent, doesn't really stand out, you can't see it. So we use stains. You can almost think of them like food coloring, okay, except they don't come out as anything. And what happens is for different stains, different structures will absorb those stains and show up better, okay? So methylene blue is a stain that we use a lot of times to be able to see some of the organelles, those structures within cells. So those are onion cells, and you see the nuclei there. Those are human sheep cells, and you see the nuclei there. Notice the different type of cell as we're going through this. Notice that the onion cell is very structured. That's a plant cell, so it has a cell wall. What's the cell wall of a plant cell made of? Chapter 3. Carbohydrate. Structural polysaccharide. Cellulose. Okay? But notice that the animal cell doesn't have that. Animal cells don't have cellulose. This is an Elodea cell. Again, this is the plant back in the aquarium back there. This is what you're going to see when you look at the Elodea in Lab 3. And notice that there is the nucleus. That is an unstained cell. Notice how clear it is. They're very, very hard to see when they're not stained. And you're actually going to be seeing Elodea cells in the microscope. The little green disc-looking things are chloroplasts. We're going to talk about those later on. And notice that they labeled the cell wall. Again, plant cells have cell walls that give them a rigid structure. Okay, we've discussed earlier in Chapter 1 that there are actually two different types of cells. Just as a little review, make sure that we're all on the same page. Remember, we said that there are prokaryotic and eukaryotic cells. We talked at length about what that meant, where those words came from. Pro meaning before, karyotic referring to the nucleus, coming from the Greek word karyos. Eukaryotic, eu prefix meaning true. Again, karyotic referring to the nucleus. So prokaryotic would be those cells without a nucleus, or those cells that came before the nucleus. Eukaryotic would be those cells with a true nucleus. This all kind of coming back to you, maybe. Okay, just to get it in your notes, make sure everybody's got it. Prokaryotic cells don't have any membrane-bound structures, and the most prominent of those is the nucleus. So prokaryotic cells do not have a nucleus. They are very, very simple when compared to eukaryotic cells. Extremely simple. They are very small. They are from 1 to 10 micrometers on average. Very, very small. That's a test question. Hint, hint. 1 to 10 micrometers. Remember micrometers, not millimeters. So bacteria are an example of that, and they are small, 1 to 10 micrometers, very simple prokaryotic cells. Eukaryotic cells, on the other hand, these were those cells that do contain organelles that are membrane bound. These are compartments within cells that have membranes around them. Okay, and each one of these has a job to do. We're going to call them organelles. Organelle, the word organelle, literally translated, means little or tiny organ. So you can almost think of these little membrane-bound structures as the organs for the cells. Just like your body has organs that perform functions, these cells have little compartments that are surrounded by membranes, and these organelles have jobs to do. Okay? And the largest one, usually in a cell, is the nucleus. So eukaryotic cells have membrane-bound structures called organelles, including a nucleus. They are very complex compared to prokaryotic cells. And they, as you can see in the illustration, they're larger than prokaryotic cells, usually between 10 and 100 micrometers. And again, this would be everything that's not a bacterial cell, most living things. Everybody okay? Okay. For the rest of the lecture, we're actually going to talk about these two types of cells, and I'm going to give you some characteristics and some structures that are included in them. We're going to start with prokaryotic, just some basic information. We have a whole chapter 20, that deals with bacteria, so we're going to save some of that for that. And then we're going to talk about eukaryotic cells and the structures within those cells. So let's start with prokaryotic. Prokaryotic come in 3 generalized shapes. Okay? They are either coccus or cocci, which is spherical. They can be rod-shaped, which is bacillus, or they can be spiral-shaped that we call spirillum. So 3 generalized shapes, and there are some variations of these, but 3 generalized shapes: round, coccus, rod-shaped is bacillus, and spiral is spirillum. Let's take a look at just a composite bacterial cell. And again, this is just an illustration. This cell has a little bit of everything. You might not find a bacteria that has all of this, but this is a composite bacterial cell. The covering of the cell we're going to call the envelope. The envelope has several different components. The first component is the plasma membrane. That's the layer closest to the cytoplasm. The membrane is always going to separate the cell, the contents of the cell, from the outside world, from the rest of everything. Outside of the plasma membrane is going to be the cell wall, and it's made out of a protein sugar substance. That's the next layer. Peptide, you learned, was protein. Glycan is sugar. That's the cell wall. Again, the cell wall of any cell, if a cell has a cell wall, the wall is always permeable to everything. It's never the cell wall's job to control or regulate entrance into the cell. The plasma membrane always does that. The next layer is called the glycocalyx. The glycocalyx is made out of polysaccharides, and that's going to be the third layer. So all three of these together we call the envelope. But the plasma membrane is always going to be that first layer that separates the cytoplasm and the contents of the cell from the rest of everything else. That's always going to be that first layer. Let's talk for just a moment about the glycocalyx, because it can take different forms depending on what type of bacteria we're looking at. If the glycocalyx has a very loose structure, and that's that outside covering that you see on the very, very outside, if it has a very, very loose covering, we call it a slime layer, and it's slimy. For those bacteria that have that type of glycocalyx, it's used to help the bacteria adhere to surfaces. They stick. Example, when you wake up in the morning and you run your tongue across your teeth and your teeth feel really slimy, that's the glycocalyx of the bacteria that have grown in your mouth overnight. I know that's kind of gross to think about, but your mouth is a wonderful place for bacteria to grow. They like moist, dark, warm areas, and that's exactly what your mouth is. So you got lots of bacteria that grew in your mouth overnight. If the glycocalyx is more structured, it takes the form of a capsule. For those bacteria that have capsules, those capsules actually help to protect the bacteria. Everybody with me? Any questions? Okay, let's move on. The cytoplasm, that fluid inside the cell. It's a semi-solid, kind of a jello-type substance, a semi-fluid solution, mostly water, at the site of lots and lots of metabolism. Lots of reactions occur in the cytoplasm. As a matter of fact, we said living things were 70 to 90% water, so somewhere between 70, 80, 90%. The cytoplasm would be water. Let's take a look at the appendages. An appendage is anything that extends beyond the body. So let's talk about those things that we see sticking out of the cell. Notice the long whip-like structure at the very bottom of that cell. That's called a flagella. It's very thin. It's there to help the cell move. It thrashes. It's long and thin. It's there for motility. It whips. The human body has a cell that has a flagella. What is it? Anybody know? You can say it. It's a sperm. It's not a four-letter word. You can say it. It's a sperm. Yeah, we have a cell that has a flagella. The other structures, the long whisker, the short whisker-looking things all over the cell, those are called fimbriae. The bristle-looking, those are for adhesion. They help the cell to adhere to surfaces. And the long tube-like structure that is extending from the left of the cell, looking at the slide, that is called a sex pili or sex pilus. That is a hollow tubular structure. You could almost think of it as like a hypodermic needle type structure. It's very rigid. And let me tell you what the sex pili of bacteria are used for. How do bacteria reproduce? Does anybody remember? They reproduce, what did we call it in chapter 1? We called it binary fission. That's exactly right. And that is simply splitting in two. So they have asexual reproduction. They literally make a copy of themselves. And that's how they reproduce. Division is how they multiply their population. When you have asexual reproduction, we discussed in chapter 1 that there's no genetic variability. If you reproduce asexually, that means all your offspring, if there's no any type of genetic change or mutation, that all of your offspring are going to be exactly like you. Well, we know that different genetic makeups have different physiologies. Just like in here, everybody in here is different, so we all have different physiologies. If everybody were the same, if we reproduced by binary fission and everybody was the same, that means that we would all have the same strengths and we would all have the same weaknesses or susceptibilities, right? So we know that some of you get the flu the minute it comes into town, right? And some of you hadn't had the flu in years. Well, if we're all genetically alike and a virus comes in the door that we're susceptible to, we may all die. That's not good for the population. Differences are good. Diversity is good for the population. It means that if something comes in that's a threat to our population, if we're all different, some of us may survive. Okay? So, organisms like bacteria that reproduce asexually, most of them are going to have some strategy within their life cycle or their natural history. Most of them are going to have some strategy to get with another individual and mix up the genes, just so that there will be some differences in the population. Now, it's not reproduction. It's just getting with another individual of your species and swapping some DNA. And these bacterial cells do that. They come together, they fuse their sex pili, and they swap pieces of DNA. So after it's over, what you end up with is two cells that are different from when they began, and then different from each other also, and you have some genetic variation in the population. Those two cells would then start binary fission and producing more like themselves. Okay? So we call it conjugation. DNA transfer is called conjugation. Okay? Let's look at some other structures within the bacterial cell that may be of interest. All bacterial cells obviously are going to have a chromosome. Bacterial cells have one chromosome, one cellular chromosome. It's circular, and it's illustrated in this illustration by this right here. It's pointing to this right here. That is the chromosome. The area where the chromosome is located is called the nucleoid. N-U-C-L-E-O-I-D. And it's just a space. It's not an organelle. It's just the area in the cytoplasm where that one chromosome is located. Don't get that mixed up with nucleus. It's not a nucleus. It's just a space. It's a nucleoid, and that's where the chromosome will be. They also have these circular structures right here. This is the ribosome. Right there, those little round structures. Ribosomes are where proteins are put together. Remember the word synthesis means to put together. That was one of your root words. So this is where the proteins are put together. Go back to chapter 3 again, and we're probably going to say that a couple of dozen times this semester. But when we're putting a protein together, remember the monomers of a protein, they were what? What's the monomer of a protein? Amino acids. That's exactly right. So the ribosome is where the amino acids are put together. And remember we discussed how important the sequence of those amino acids were. that specific proteins have a specific sequence of amino acids. So this is where that is determined. And we're going to talk in chapter 12 about how that works. How does the ribosome know what order to put the amino acids in to make a protein? But for now, just know that the ribosomes are the site where those amino acids are put together to make proteins. So this is where proteins are synthesized. Some bacteria have what's called a thylakoid. Thylakoids contain chlorophyll. It would look something like that. I want you to start associating the word thylakoid with chlorophyll. Okay, what other organism has chlorophyll that you're really familiar with? Come on, guys. Plants. That's exactly right. We're going to talk about thylakoids again when we talk about plants. I want you to associate thylakoids with chlorophyll. So, chlorophyll is where photosynthesis takes place, or part of photosynthesis takes place. So, if you have a bacteria that has thylakoids, what can it do? It can photosynthesize. It can make its own food. So these bacteria that have thylakoids are autotrophic bacteria. Okay? We commonly refer to them as cyanobacteria. That's a word you need to know. Those are autotrophic bacteria. What does that mean? That means they make their own food. Very important. Okay, guys, when you were growing up, some of you had the 64 box of Crayola crayons, right? Who had the 64 box? Show me. You privileged elite. You guys had 64. The rest of us had 8 and 16, okay? I'm just kidding. But you had 64. You're the guys that had the sharpener in the back of the box, right? Yes, you had the sharpener. But if you had 64 pack of Crayola crayons, you had a crayon called cyan. Does anybody remember that crayon? Yes, what color was it? It was blue. It's a blue-green color. Okay, so these bacteria are a blue-green color. The chlorophyll causes them to be that color. Okay, they're called cyan bacteria or cyanobacteria. They're blue-green. They're photoautotrophic. They actually can photosynthesize. And I'm just kidding about the 64. Okay. Some bacteria have additional pieces of DNA called plasmids. Okay? A plasmid is not the cellular DNA. A plasmid would be another circular piece of DNA that would be within the cell. Okay? But it's not the cellular DNA. It's additional DNA. Let's just go ahead right now and lay the framework for a couple of chapters from now. And let's say that DNA is the recipe to make a protein. DNA tells the cell the sequence of amino acids in a protein, the recipe, what order they're supposed to be in. And proteins actually give the cell properties or characteristics. Okay? So, if we've got additional pieces of DNA, then we have other things that are being made. This is not cellular DNA. So we have other proteins that are giving those bacteria some properties or characteristics that maybe other bacterial cells don't have. Okay? These plasmids are not cellular DNA. The cell doesn't have to have them to survive. As a matter of fact, technologically, listen to this for just a moment. We just defined DNA, the area of the gene specifically determines the sequence of amino acids in a protein. Well, we know that insulin is a protein, right? Insulin is a protein. We have figured out how to, we know where the human insulin gene is. We know in your DNA where the gene is, the recipe to make insulin. So we have figured out how to take these plasmids out of bacterial cells. Remember, the bacteria, it's not the cellular DNA, so they can do without it. We take the plasmids out of the bacterial cells. We open it up in a specific place. We take our human insulin gene from a human cell, put the human insulin gene within the plasmid, close it back up so that it's complete again, and we can take that plasmid and put it back in a bacterial cell, and that bacterial cell will produce human insulin. Amazing technology. Revolutionary for diabetics, because I think historically diabetics have had to use swine insulin. Anybody in here know? I think it's swine, the insulin that they have to use, which in and of itself, because you're using insulin from another species, causes some health problems in those people. So the fact that we now know how to harvest human insulin from bacteria is a tremendous breakthrough for diabetics. Some bacteria have endospores. An endospore is a dormant condition for a bacterial cell. For those bacteria that can produce endospores, when they come upon a harsh environment, when conditions get bad, when it looks like that their survival might be threatened, they'll produce these endospores and go dormant, and they can withstand long periods of time in really harsh conditions and even be dispersed by elements or other organisms to a better environment. And then when the environment gets better, they'll produce bacteria again. They'll be viable bacteria. Some bacteria that produce endospores that you may be familiar with, botulism produces endospores. Anybody know what botulism is? If you've ever had it, you will never forget what botulism is. Anybody ever had botulism? Food poisoning. This is why you have to heat your food up high when you're canning, because that bacteria actually can produce endospores that can survive extreme heats and live for long periods of time, and then make you sick when you open the can back up. That is an endospore. Okay, let's take a look at some of the differences between prokaryotic and eukaryotic, some of the things they have in common. You see there on the right side, prokaryotic cells, as we have discussed, have no nucleus, no membrane-bound organelles. They are very primitive, and note that word right there. We haven't talked about this, but we're going to. Prokaryotic cells do not have a cytoskeleton. And obviously this is not like your skeleton. It's mainly protein fibers, but it is a structure that helps to give the cell form, and prokaryotic cells do not have that. On the other side, eukaryotic cells, we're going to talk about now, have a nucleus, they have membrane-bound organelles, they are more complex, and they do have a cytoskeleton. And again, we're going to talk about that toward the end of the lecture. What I wanted you to see here was what they have in common. They both have cytoplasm, and it's very similar. It's in both types of cells. It's primarily water, but it is the site of lots and lots of cellular reactions. They both have DNA. Every cell has DNA. They both have plasma membranes, and they both have ribosomes. And again, ribosomes were the site where proteins were put together. Okay, so let's move on to eukaryotic cells. Again, this would be all except bacteria. So we're talking about the cells of animals, plants, fungi, protists. Those cells that have a membrane-bound nucleus. We see the nucleus right there. It's usually the most prominent structure seen in the cell. There's the nucleus. Lots of organelles. That's all these structures we see that are labeled here. We're going to go over them and have a general understanding today of what all of these things do. So let's first take a look at something you should be familiar with, which is the membrane. We've talked about the membrane, and we've said that it separates the contents of the cell from the outside, and that it regulates the passage of molecules inside and outside the cell, into and out of the cell. You should be very familiar with the membrane. We've talked about how it's made of those phospholipids that form a bilayer in the presence of water. You guys should be familiar with the fact that the head of that molecule is what? The head is hydrophilic and the tails are hydrophobic, yes, and it does form that double membrane in the presence of water. Okay? The cytoplasm, as with the bacteria, is primarily water. The fluid of the cytoplasm we call cytosol. And again, we're going to talk about the cytoskeleton in just a moment, but the cytoskeleton would be those things right there that look kind of like noodles in the illustration. That's parts of the cytoskeleton. Here's one up here, right there. That's part of the cytoskeleton, that actin filament. But all together, the fluid and the filaments we call the cytoplasm. The ribosomes, again, we said are the site of protein synthesis. In a eukaryotic cell, we find them in two different places. We find ribosomes attached to another organelle, and that organelle is right here. It's right here. They're attached to the endoplasmic reticulum. And as you can see right here, the ribosome is actually a two-unit structure. There is a large unit and a small unit that comes together to form the ribosome when proteins need to be made. But we see it attached to the endoplasmic reticulum. There's two types of endoplasmic reticulum. We're going to talk about that in just a moment. This portion right here, which is closest to the nucleus, actually has the ribosomes on it. And if you'll notice the little bumpy things right there that make it look kind of rough or studded, okay, because the ribosomes are attached to that structure, we're going to call that the rough endoplasmic reticulum, as opposed to this area out here that does not have ribosomes. We're going to call that the smooth endoplasmic reticulum because it doesn't have ribosomes on it. It looks smooth. Okay, so we see those attached to both the ER right here, and we can also find them free floating in the cytoplasm. There's some right there. Those are free floating in the cytoplasm. So we find ribosomes in two places in eukaryotic cells. We find them attached to the ER, up closest to the nucleus, and we find them in the cytoplasm. But wherever they are found, they are the site of protein synthesis. The nucleus, the control center of the cell. We see this eukaryotic cell right here. The large structure is the nucleus, that right there. And they've already cut a portion out of it so you could actually see the inside. But this area of the cell controls everything about the cell. It controls when the cell divides. And we're calling that reproduction, basically. So it's going to control the reproduction of the cell, splitting in two. It stores the DNA, the chromosomes, so it stores the genetic material. And it controls the structure and the function of the cell. So it controls everything about the cell. So let's take a look at this nucleus right here. and talk about some of the things that we see inside the nucleus. Notice that there's this really dark area right there. We'll see that, and then you see outside that dark area there's some light places right there, and then there's some shadows. Do y'all see all those shadows that exist out in the, this is called nucleoplasm. You see those shadows right there? Let's talk about what those are. Those light places, right, the dark places in between the light places, the shadows right there. The shadows, if you see this word right here, the shadows are areas of chromatin. And chromatin is your DNA in a non-dividing cell. Your DNA in a non-dividing cell. Let's talk about that for just a moment. I'm going to go ahead and set the stage for chapters in the future as we talk about that. Take a few moments here to kind of set things up. In your cells, and when I say a non-dividing cell, do you guys remember a while ago when we talked about cells being small and growing and growing and growing and getting big and having to divide? So basically when we say a non-dividing cell, we're talking about that cell that has just come out of division up to the point where it divides. That period of time where it's growing. Okay? So in a non-dividing cell, before division, in those cells, your DNA exists as chromatin. And I'm going to draw lots of strands. Okay? Let's just say this is chromatin. And we can't see that with a compound light microscope. You can't see chromatin. That's why in the illustration it was shown as just a shadow. Because chromatin is really, really diffuse. That means it's loose and strung out. Okay? But if I had an electron microscope and I looked deep, deep, deep into that piece of chromatin, I would eventually see way down in there the double helix structure that we know is DNA. Okay? So your chromatin is DNA with some associated proteins that are keeping it wrapped up, but that's your DNA in there. Okay? It's really diffuse and you can't see it in a non-dividing cell. So in a human cell, you have 46 pieces of chromatin. And they're all different lengths. There are going to be some short ones and some really long ones. Okay? But your cells have to have those 46 pieces. Every cell, except your red blood cells, every cell has to have 46 pieces of chromatin. That's what controls the cell. That's the recipes for everything that the cell needs to make. Okay? So, the cell is growing. It's growing and growing. And eventually, we saw a while ago, eventually that cell is going to get to a point where it has to divide to stay alive, right? Right? So every cell needs 46 pieces of chromatin. Before the cell even gets ready to divide, it makes a copy of every piece of chromatin and hangs on to it, okay? So it's going to make a copy of that one and it hangs on to it. It's going to make a copy of that one and it hangs on to it. It makes a copy of every piece of chromatin. So now, once that's done, and that's called replication, okay, now that it's made a copy, now really there's 92 pieces of chromatin, right? Because every one of the 46 made a copy of itself. So there's two sets. They're just alike, right? So now the cell continues to grow and it's getting closer to the time of division. So the cell knows eventually it's going to have to put each one of those copies into another cell. So those are literally going to be torn apart and put into cells. 46 on one cell and the other 46 on the other cell, right? They're going to split the copies. Well, they're really, really long and stringy, and they're hard to control when they're long and diffuse. So let me ask you a question. If you were going to move, if I'm going to move these chromosomes, split them in half, and put half of it in one cell and half of it in another cell, what do you do right before you move? What do you do? What do you do with your stuff? What do you do with it? You pack it up. That's exactly right, because stuff is a whole lot easier to move when it's condensed into a small container, right? So these chromosomes literally condense. I like to think of it as the curly ribbon that you use at Christmas. You know, I'm talking about the curly ribbon that you start out with like this 6-foot long piece of ribbon, and you take a pair of scissors and you go, and that 6-foot long piece of ribbon goes, and now it's like 3 inches long. You know what I'm talking about? I mean, it's the same 6 feet. You've just condensed it into 3 inches, right? You know what I'm talking about? That curly ribbon. These chromosomes do exactly the same thing. So if I'm just going to do one of them. Is it okay if I just do one instead of all 46? Yeah. I'm going to take one piece of chromatin that has replicated itself, okay? And that right there, what's holding those two copies together, we're going to call that the centromere. Centromeres actually hold the copies together, okay? Well, after that's replication, now we're going to condense it so that one side does this and the other side is going, and now that is the centromere. Okay, same thing. It's exactly the same thing. It's just condensed now. Okay? Most books illustrate that like this. What is that? That's a really bad drawing. Okay, guys? I will redo that. Most books illustrate a condensed, replicated piece of chromatin as that. What is that? That's a chromosome. That's exactly what that is. That's a chromosome. So from now on, when you see chromosome and you think about what a chromosome is, realize that that's a piece of chromatin that has replicated itself and condensed. So genetically, there's enough information in that chromosome for 2 cells. Whatever recipes was on that piece of chromatin, now there's 2 copies of them. Because that is going to be split into 2 cells. So that each cell would have 46 pieces of chromatin. You got it? Is everybody okay? Any questions about chromatin, chromosomes, replication, condensing? Okay, let's move on. So your chromatin is your DNA in a non-dividing cell. Chromosomes would be chromatin that has replicated and condensed, and we can only see chromosomes in dividing cells. We can only see DNA when your chromatin is condensed. So whenever you see a picture of somebody's DNA, whenever you see a picture of a chromosome, know that that cell that chromosome was in had to be going through division, because that's when they condense, right before division. Okay, the nucleolus is that dark spot in the middle of the nucleus we see right there. That's the nucleolus. That is the place where the ribosomal subunits are made. Those 2 subunits that come together to produce the ribosome. The nuclear envelope, if we were to cut this area right here off the nucleus and take a look at it really close, it would look like this. You would see that the envelope, or the covering over that organelle, that nucleus, is actually a double membrane right here. There's a double membrane that kind of folds back on itself. And there are these pores right here that allow molecules to go in and out of the nucleus. If they can fit through that hole, they can go in and out of the nucleus. Your DNA cannot leave the nucleus. It's too big a molecule. It can't leave. It can't go through those pores. And that's a good thing. Your DNA is the master copy of all the recipes that the cell needs. So you don't want it out of the nucleus. The cell has a system. Just like your body has systems. And we talked in chapter 1 that a system, an organ system, was made of organs that come together for a common function. Well, just like your body has a system, the cell has a system. It's called the endomembrane system. And there are actually 4 organelles that participate. The first one is the ER, or endoplasmic reticulum. And again, remember there's 2 different types of that. The ER participates in this system. Something called the Golgi apparatus participates in the endomembrane system. Lysosomes participate, and vesicles participate in the endomembrane system. So these 4 organelles participate in this system. So before we get into what each one of them does, let's talk about what the system is. What's its function? What is the endomembrane system? Well, you can see by looking at all those organelles that participate that it's basically just a whole lot of sacs and channels. Lots of sacs, lots of channels. So what does it do? What's the function of this? The purpose of these 4 organelles, and the reason it's called endomembrane is because all 4 of these organelles are covered with the same membrane. It's the same kind of membrane. They are either physically connected or they connect by vesicle transfer. So the membranes are all the same. But what do the 4 of them do? They literally synthesize and transport all types of molecules. Synthesize and transport molecules. Remember the remember the 4 classes of organic molecules, the carbohydrates, the proteins, the nucleic acids, the lipids. The cell's going to be making those. Some of those molecules the cell itself might need. Some of those molecules may need to be moved out of the cell, taken out of the cell for other purposes. But this system is responsible for synthesizing and transporting them. That's just a fancy way. If you want to mark out those 2 words, that is just a fancy way of saying this system makes and moves molecules. It makes and moves them where they need to be. Again, the cell's making proteins. Some of the proteins may need to stay on the endoplasmic reticulum. Some may need to go to the cytoplasm. Some of the proteins may need to go to the membrane, the plasma membrane, and stay there. Some of the proteins, like insulin, the cells that make insulin actually transport it out of the cell. So some of these molecules have to be moved around. It also, second thing the system does is it restricts enzyme activity to certain areas. So there are some reactions that need to be compartmentalized. It restricts enzyme reactions to certain areas. Okay, let's start with the endoplasmic reticulum. There are 2 types, as we mentioned earlier. They're first closest to the nucleus right here, and you can see it better in this illustration. You can see these little circles that are attached to these channels right here that makes it look kind of rough. That is the rough ER. It has ribosomes, and again, ribosomes are where proteins are put together. So we associate the rough ER with protein synthesis. The smooth ER, which would be on the outside of that structure, it would be this right here, this area that is still endoplasmic reticulum, but it doesn't have ribosomes on it, so it looks smooth. It's right here. The smooth ER actually synthesizes other types of molecules. Excuse me. A good example of that would be lipids. Lipids are made in the smooth ER. So the molecules are made. And again, some of them need to stay there, some of them need to go to the cytoplasm, some of them they need to go out of the cell. But it's not very efficient just to make a molecule and move it by itself. So to be more efficient, the cell literally has a distribution center. You could almost think about the endomembrane system like the Walmart Corporation. Okay, Walmart has a distribution center, right? Where everything goes to the distribution center, and then the distribution center packs up trucks to send them to stores, right? So the cell literally manufactures molecules and sends everything to the Golgi. The Golgi is responsible for accepting those molecules, processing them, packaging them all together, and distributing them. So the Golgi may package several different types of molecules together that need to go out of the cell. Instead of just sending each one of them individually, the Golgi improves the efficiency of the whole system. And it looks like pancakes. It always helps me to think of something as a food item. I can remember things if I associate it with food. So how do we get it from the ER to the Golgi? If this were the Walmart system, we're missing the 18-wheelers, right? What's moving the molecules from one place to another? That's the vesicles. The vesicles are just little sacs that transport molecules. These are the little sacs right there that transport large molecules. Right there's a vesicle. All these little sacs would be vesicles. There's a vesicle that we need to talk about. It's a very special vesicle. It's specialized. It's called a lysosome. What does lysis mean? Root word. What is lysis? This one's an important one. To break or to burst. Okay? Lysis. To break or to burst. So these lysosomes, what do you think they're doing? They're breaking down molecules. This is where digestion occurs. Lysosomes are involved in digestion. Breaking down molecules. Think about it for a moment, guys. Cells have to break down molecules, just like your body has to break down molecules. You have a stomach that breaks down molecules for you, right? You have enzymes within that compartment that we call your stomach that literally breaks down molecules, carbohydrates, lipids, nucleic acids, proteins, etc. Keep in mind, all living things are made of the same molecules. So when you eat a hamburger or you eat plants, it doesn't matter what you're eating. Those things that you're eating and digesting are made of the same molecules that you're made of. So literally those enzymes within your stomach could digest you. That's why it has to be compartmentalized. If you don't compartmentalize digestion, you digest yourself. Okay? I'll never forget, I said that about 7 years ago to a class, and a little girl on this side of the room burst into tears. And I mean, she wasn't just quietly crying, she was sobbing. And the whole class turned and looked, and I said, I'm so sorry, are you okay? And she said, I'm fine, I'm so sorry. She said, I'm so sorry, but that just happened to my brother. And I said, what? And she said, he just died. And I said, what do you mean? And she said he had an ulcer in his stomach, and he didn't know he had an ulcer, and it ate through the lining of his stomach. And before he realized what was going on and they could get him to the hospital, the digestive enzymes had seeped into his abdominal cavity, and it killed him. It digested him. And he died. So that's exactly what had happened to him. Lysosomes are also involved in something called apoptosis, which is programmed cell death. Apoptosis. Your cells are designed to keep you healthy. Given half a chance, your cells want to be healthy. You have molecules that constantly scan your DNA to make sure that everything's okay. And if something happens and you end up with some type of mutation or change that's not right, and these molecules are there to recognize it, and some of them are called proofreaders. And they're proofreading everything. They want you to be healthy. They want DNA to be right. And if something happens, they literally can change and fix DNA. So when something's not right and they can't fix DNA or they can't fix your cell is designed to commit suicide. And that's what apoptosis is. It's cell suicide, programmed cell death. They are programmed to destroy themselves under certain conditions. If it can't be fixed, it's better for one cell to be destroyed than for that cell to divide and you end up with two cells that have a problem. And then those two cells divide and you end up with four cells that have a problem. And then 8 cells, and then you end up with cancer. And that's what cancer is. Cancer is uncontrolled cell division. Okay, we're going to talk a whole lot about that in chapter 9. But your cells are designed to try to avoid that. So you should be thinking right now and asking me a really obvious question. If my cells are designed to self-destruct if there's a problem, then why do we have problems? Why do we have cancer? If cells are designed to kill themselves, to commit suicide when there's a genetic problem, then why do we have cancer? Well, I want you to think about it for a moment, because those molecules that are monitoring your cells, those molecules that we call proofreaders that are proofreading your DNA to make sure that it's healthy, those are enzymes. Okay? Do you guys remember what an enzyme is? What type of molecule is it? Where did we talk about enzymes? In chapter 3. Enzymes are proteins. Okay? So the molecules that are keeping your cells healthy are proteins. Where's the recipe to make that protein? Where's the recipe to make a protein? In the DNA. So the molecule that's keeping the cell healthy is a protein, and the recipe the cell used to make that protein is in the very DNA that it's monitoring. So it's almost a catch-22. The molecule that's keeping your cells healthy by monitoring the DNA is coded for in the very DNA that it's monitoring. So if that gene that is the recipe to make that proofreader gets mutated, then you don't have that. Okay. We'll talk more about that in chapter 9. There it is. Is it not beautiful? Look at that. That is the endomembrane system in totality. So let's just summarize it really quickly. What is this right here? What is that area? What's it called? The rough ER, the rough endoplasmic reticulum. What are those little red things right there? Ribosomes. So notice how this right here is budding off the rough ER. Literally is budding. So it turns into a vesicle when it buds off. So what I want you to recognize here is the membrane that is surrounding that vesicle used to be part of the ER. It just budded off the ER. So all of these membranes are recycled between these four organelles. Okay, so that vesicle is traveling to the Golgi down here, and notice how it just kind of bleeds into the Golgi. So the membrane that was the vesicle is now the membrane of the Golgi, and those molecules that the vesicle contained right there are now being deposited into the Golgi. So if it's coming from the rough ER, what type of molecules would those be? What does the rough ER make? It has ribosomes, so it makes proteins. So those are proteins. They're taken to the Golgi right here. The Golgi is processing them and redistributing them. Notice how this vesicle is now budded off the Golgi and is moving toward the plasma membrane. So that's the plasma membrane. And that vesicle just blends in with the plasma membrane and becomes part of the membrane right there as the molecules are released to the outside of the cell. That is called exocytosis. That's leaving the cell by vesicle. Here's a molecule right here that needs to come into the cell. This is vesicle formation. That's called endocytosis. When you're bringing things into the cell by vesicle formation. It's traveling up through the cytoplasm, and I don't know what that molecule right there is yet, but when I see that it merges with my vesicle, and then I look at the next step, and I notice that those molecules right there are now smaller, what was that? Whatever was in that has broken down my molecules. Whatever that is caused those brown molecules to be smaller. So that's got to be some type of digestion. So then this is a lysosome. Okay? So that is the endomembrane system right there. Couple of other organelles that do not participate in the endomembrane system but are very, very similar. Peroxisomes are specialized vesicles, very similar to lysosomes. They contain enzymes, but they are very cell-specific. They usually associate peroxisomes with the liver and hydrogen peroxide. This organelle right here, this is a plant cell, you can tell, because it has this rigid cell wall, also has the chloroplast right there. But this giant organelle right there is called a vacuole. That's a sac, a lot larger than a vesicle, huge usually, and they are used to store substances. Vacuoles in plant cells, that one right there, every plant cell has a central vacuole. And the central vacuole is large. It stores water for the plant cell. Okay, a couple of other organelles that are really, really important. Let's talk about energy in the cell. These are energy-related organelles. Chloroplast, an energy organelle, is found in plant cells. And mitochondria, another organelle associated with energy, is found in plant and animal cells. So let's take a look at the chloroplast first. Chloroplast. Here's a chloroplast. This is the organelle. They've cut a piece of it out right here. It looks kind of like a green Skittle, but they've cut this piece out. This organelle is where photosynthesis takes place. That's where we're going to convert solar energy to chemical energy. This is where the chlorophyll is found for photosynthesizing. Let's back up just a minute. Let's make sure we understand the words that are on that screen. We're taking solar energy and we're converting it to chemical energy. What is chemical energy? We've talked about energy a little bit in chapter 2. We learned that how do we store energy chemically? It takes energy to form these, energy is stored in these, and energy is released when these are broken. Bonds. Okay? Chemical energy is the energy that's stored in bonds. So if we're taking solar energy and we're converting it to chemical energy, what are we doing? We're bonding something together. We're taking solar energy and we're bonding atoms together and storing that energy in a molecule. We're building a molecule. That's what chemical energy is. We are building a molecule. Okay, that's what photosynthesis is. So let's take a look at the organelle. I've got a whole chapter devoted to photosynthesis. We're going to talk about the organelle right now. It's a double membrane organelle. It has an inner membrane and an outer membrane right there. Inside the organelle, we have these stacks of right here. The stack is called, the whole thing right there is called grana or granum. And each individual little unit right there is called a thylakoid. And there's fluid in those thylakoids, and that is the chlorophyll. So the whole thing, that whole thing is called granum or grana. Each individual thing is called a thylakoid. So there's stacks of thylakoids right there. And within each thylakoid you would find chlorophyll. There is a fluid that surrounds, this is a fluid right here. There's a fluid that surrounds the grana. It's called stroma. And there are lots and lots of enzymes right here for photosynthesis. Lots of enzymes in the stroma. And these would be called the thylakoid spaces where the chlorophyll is. Remember the word thylakoid? We talked about it when we talked about bacteria. Thylakoids contain chlorophyll, and if you have bacteria that has thylakoid, it's autotrophic. Remember that? Short-term memory's failing you. Okay, I'm just kidding you. Okay, so what do we have here? We have chloroplast, double membrane. The whole thing right here is called grana. Each individual one is a thylakoid. That's where the chlorophyll is. The fluid surrounding the grana is called stroma, and lots of enzymes. Half of photosynthesis takes place in the grana, half of photosynthesis takes place in the stroma. We'll talk about that later. Chloroplasts are a little bit unique in that this organelle has its own DNA and its own ribosomes. And that's really different. Other organelles don't have that. It has its own DNA and its own ribosomes. What do I mean by that? That's not cellular DNA. That DNA contains the recipes for proteins that only the chloroplast needs. So it has its own recipes, has its own DNA, and it has its own factory. It has ribosomes. So not only does it have its own recipes, it can make its own proteins. Okay? And they can reproduce themselves without the cell, which is really unique. Okay? Mitochondria, site of cellular respiration. We sometimes call this the powerhouse of the cell. The powerhouse of the cell. Anybody ever heard of cellular respiration? Have you heard of that before? High school biology, maybe? No, maybe not. Okay, I'm going to give you cellular respiration in a nutshell. Listen to me carefully, because I find that most students, even if you had it in high school biology, you come away not really understanding what's going on. We call it the powerhouse of the cell, because this is where the cell gets its energy. Remember we talked earlier, again in chapter 3, how many times am I going to say that? Remember we talked about ATP? Y'all remember ATP? We said that when a cell needs energy, it has to get it from ATP. That's the energy a cell uses. That's the energy a cell understands. That's the only energy a cell uses. Okay? So, what's this cellular respiration thing? Well, when you consume food, let's take carbohydrates for instance. When you consume carbohydrates, your body breaks it down into the monomers of that molecule. What's the monomer of a carbohydrate? A one sugar, which is monosaccharide, okay? So your body gives the cell a monosaccharide. It gives it glucose. Your body says, okay, cell, here's you some glucose. Well, the cell has no idea what to do with glucose. Glucose, it doesn't understand it. It's way too much energy. It's way too much. It would be like you taking $100 bill to the Coke machine. What's going to happen when you put the $100 bill in the Coke machine? It doesn't understand it. It's too much, right? So what do you have to do with that $100 bill if you want a Coke? You have to go to a change machine, right? And you put the $100 bill in a change machine, and out comes $100 worth of ones, right? And now the Coke machine understands the ones, right? Well, when the body gives the cell glucose, the cell says, I don't understand glucose. That's too much, just like the $100 bill. Glucose is a stick of dynamite when the cell really wanted firecrackers. Okay? So the mitochondria takes the glucose, and the mitochondria is the change machine. The mitochondria takes the glucose and spits out 36 ATPs. For every glucose molecule, the mitochondria gives 36 ATPs. Just like for every $100 bill, the change machine gives 100 ones. And the cell then can use the ATPs. Okay, glucose goes in, 36 ATPs come out, the cell uses the ATPs. Okay? So that's what cellular respiration is. That's why we call it the powerhouse of the cell, because it takes glucose and turns it into a form, ATP, that the cell understands. Okay? And it's that peanut-shaped organelle right there. Let's take a look at it. That's the mitochondria. It, too, is a double-membraned organelle right there. Except, unlike the chloroplast, the inner membrane of the mitochondria actually folds on itself right there. Those little folds, those are called cristae. And this is where most of the ATPs are formed, right there. So the cristae right here is ATP production. You guys remember ADP plus P yields ATP, right? With energy goes in, ATP comes out. The space right here in the middle, this is called the matrix, and it has a fluid there, again, the site of lots of reactions. That's the matrix. And the mitochondria also has its own DNA and its own ribosomes. So what do I mean by that? I mean the mitochondria has the recipes for proteins that only it needs, and it has the ribosomes to put those proteins together. Anybody ever heard of mitochondrial DNA? A lot of times when you're tracing your ancestry, we use mitochondrial DNA instead of cellular DNA because it's easier, it's faster. Think about it for a moment. Let's step back. I want you to learn something today. Anybody interested in ancestry? Uh, when a sperm fertilizes an egg, we're going to see very soon that the egg is giant compared to the size of the sperm. So the egg contains lots and lots of organelles. The sperm contains very few. So when a zygote is formed, an egg and a sperm come together, most of the organelles for that resulting zygote are going to be from the egg, not the sperm. So if mitochondria have their own DNA, then most of the mitochondria for that zygote is going to have come from the egg, not the sperm. So you have your mother's mitochondrial DNA. She has her mother's mitochondrial DNA. She has her mother's mitochondrial DNA, and so forth and so on. So mitochondrial DNA is traced through the maternal linkage. There actually is a book out. Some scientists, I have it in my office, some scientist has analyzed mitochondrial DNA and traced the entire human race back to one woman who had 7 daughters. Interesting reading. Anyway, they can also reproduce themselves. I tell you these things about the chloroplast and the mitochondria because some scientists believe in what's called the endosymbiont theory. Some believe that when cells were evolving on the planet millions and millions and millions of years ago, that the mitochondria were actually aerobic bacteria and that the chloroplasts were actually photosynthetic bacteria that were taken into a cell, and it was a mutualistic relationship, so they stayed. Some scientists believe that because if you look at what bacteria have cellularly and you compare it to what the chloroplast and the mitochondria have, they both have their own DNA. They both have ribosomes. They both have basically the same things, and they can reproduce themselves. Okay, the cytoskeleton, last but not least. This is the part of the cell that actually gives the cell some organization. Organelles are not just floating around in the cytoplasm. There's a lot of structure. They're anchored. There's shape involved. This cytoskeleton actually gives the cell shape. And when the organelles do move, and they do move, they actually move because of these filaments. Obviously, this skeleton is not like yours. This skeleton is a series of filaments, of protein filaments and fibers. There's 3 different types. There's one called actin. Here's the intermediate filaments, and the microtubules are right there. Let's look at each one of these just briefly. Let's start with the microfilaments. This is the thinnest of all of them. It looks basically like this. It's rod-like in structure. This is actin. This protein is called actin. For those of you that have had some anatomy in high school, you studied myosin, actin, myosin of muscles. It's the same actin. It's involved in movement. Actin right there. It enables those cells that move to move, and it allows the cell to change shape. The second type of filament is called, this is your actin right here. This is the plasma membrane on one side of the cell. This is the plasma membrane on the other side of the cell. Obviously, that is a mitochondria. These are showing you how the organelles, there's the endoplasmic reticulum right there. That's the rough ER. Those are ribosomes. It's just showing you that these organelles are not just floating around, but they are actually anchored with these filaments. The intermediate filaments are only found in certain tissues of animals. That's this one right here. Looks like a rope, very much like a rope. These are fibrous, these are proteins that are joined to form this rope-like structure. It gives the cell some internal structure, and you can see right here that it's anchoring the organelles in place. These intermediate filaments are anchoring the organelles. And the third type of filament, we're going to talk more about this one later. It's called a microtubule. It has this basic structure right here. It's these long hollow tubes. Notice that this tube is hollow. These are tubulin proteins, globular-like proteins right here that form this microtubule. These actually are like a scaffold for the organelles. They not only anchor the organelles, but I want you to think about the microtubules as being, do y'all know what a scaffold is? Have you ever seen a painter or someone working on the side of a building and on a scaffold? You know, he has to stay on that scaffold. Well, these organelles, literally, when they move, they have to stay on these filaments. These are the scaffolds. This is how they move. These are also involved in moving those chromosomes around during cell division. We talked about our chromatin replicating and condensing for the purpose of putting each piece into a different cell. The microtubules are very much involved in that, tearing those chromosomes apart and moving them into two different cells, moving the chromatin. Cilia, we haven't talked about cilia yet, but we have talked about flagella. These are structures on cells that help them to move, and they are both made out of microtubules. It would look very much like this right here. When you see, here's a flagella on the sperm. These are cilia. A place in your body where you have cilia. You have cilia in your lining your bronchial passages that are constantly moving. That cilia is beating upwards, so that if you get anything in your bronchial passages or tubes, that cilia is working it upward toward your throat so that you could spit it out. Okay, so the cilia in your bronchia constantly move. But they are designed, basically all that a cilia or a flagella is 9 of these microtubules with 2 in the center surrounded by a membrane. So that's the basic structure of a flagella or a cilia. It's a 9-to-2 arrangement of microtubules. Okay, there it is. Just some examples of animal plants. We hadn't talked about fungus much yet. We will do that later on. But at this point, you should be able to look at any cell drawing and pick out the basic structures. You should be able to identify the nucleus in any cell, plant or animal, and again, we haven't done fungal yet. You should be able to pick out the endoplasmic reticulum right here and here, smooth and rough. You should be able to pick out mitochondria, a Golgi, the chloroplast right here. You should be able to identify some basic structures in a cell. What do plant cells have that animal cells don't have? Just in comparison, plant cells have chloroplast, which were those green skittle-looking things. Here's one right here, there's one right there, and there's one. They also have this giant central vacuole right there for water storage. That's where a plant cell stores its water. And they're always going to have the cell wall right here that's made out of cellulose. We've talked about several cells that have cell walls. We talked about plant cells that have cell walls. We talked about fungal cells. What did we say the cell wall of fungal cells was made of? Okay, yes. And what about bacteria? That peptide sugar substance, peptidoglycan. Okay. Animal cells, what do animal cells have that plant cells don't have? They have something called a centriole. The centriole is used for cell division. We'll talk more about the centriole when we go into chapter 9 and 10. It actually produces the microtubules that move the chromosomes right there. Just some notes that you might be interested in, just some things I took out of other books. Again, if I were you, I would take a look at some cells and see if you could identify certain structures within those cells. And there's your Elidea cells.